Hi, everybody. This is Jeff Yates, Senior Director of Engagement for TU. Today, we have a really special topic and some great partners to talk about the issue of 6-ppdq and its impacts on salmon and trout across the country. I was first made aware of this issue actually by a member of my own chapter, Rob Popham, who is the author of the most recent article in Trout magazine that just came out on this issue. And he was so concerned about it, reading about the work coming out of Washington, that he led a series of talks locally at the chapters. And then we ran a national kind of webinar to raise this issue. Today's talk and that talk, they won't be the first or the last that we do. We're really committed to moving forward this issue and continuing to have those conversations. All of you today will receive an email with a recording, as well as those who are not able to attend but did register. Please feel free to forward that on to your friends and colleagues. We'll also keep this list of folks who are registered updated as we have more events and activities like this, more opportunities to engage on the issue. And for those of you who are TU staff or volunteers or members of the general public, I would encourage you to reach out to your state council or local TU chapter and offer to serve on their advocacy and their science committees, helping kind of connect not just with issues around 6-ppdq, but other water quality issues, other issues where we're going to be advocating for the care and recovery of salmon and trout across the country. So thank you for spending middle of your day with us. And with that, I'll pass it on to Dr. Helen Neville to do formal introductions.
Oh, all right. Thanks, Jeff. Hi, everybody. I'm Helen Neville. I'm the senior scientist for Trout Unlimited, based out of Boise, Idaho. And I just wanted to say a quick note of appreciation for Jeff, who really has been pushing on this issue, and I think connecting a lot of us across the country, not just in the West, but back in the East now, partly led and inspired by his own chapter there. So thank you all for providing the resources and getting the Trout article done. I think this is just a huge issue that we're really sort of catching up on. So I'm very appreciative to have Dr. John Hansen here today. He is a research immunologist at USGS at their Western Fisheries Research Center in Seattle. And I actually don't personally know John, so I'm even more appreciative of his willingness to hop on here. We were connected through some conversations with Jeff and other folks at USGS, who is always a deep partner of ours. And John graciously offered to give a webinar for us. So with that, I won't take up any more time, but John, just thank you for offering your time and expertise today, and we'll let you go.
All right, well, thank you, Helen. I am not going to be looking at the chat until after the presentation. So if there are questions that come up during the middle, we'll get to them after the presentation. So today I'm going to be talking about something that we've been actively involved with for going on 4 years. And that is really about tire wear particles, or tire dust, as some people like to call it, and fish health. As Helen said, I'm a research immunologist here at the Western Fisheries Research Center in Seattle, Washington, where we are the disease headquarters for the West Coast, where we primarily look at host pathogen interactions within my unit. Today, though, I'm going to be talking about something that was brought to light just recently in 2021 by Ed Kaloje's group. So I will be going over the findings of the causative agent of what is now referred to as URMS, or Urban Runoff Mortality Syndrome. The culprit is 6-ppdq, or 6-ppd-quinone. I'll be referring to that throughout the talk. Today I'll cover the species susceptibility and efforts by the USGS, including effects on invertebrates, helping to establish the aquatic water criteria with the EPA, a national mapping project, modes of action, and finally, some of our work that we are actively involved in and trying to find solutions to this problem. But first, just want to go into a little bit of a history of how long this problem has been noticed and how the discovery by Ed Kaloje's group came about. So this really started with citizen scientists and researchers back in the late 1990s here in the Puget Sound metro area in and around Seattle. And at the time, there was a noticeable occurrence of what was called pre-spawn mortality of adult coho salmon. And this occurred primarily, or at least it was noticed primarily, during the fall storm seasons that call salmon in for spawning. part of the reason why it was more visualized and this really came to the forefront was restoration efforts through Seattle, mainly removal of culverts and other fish blockages, development of more public green spaces, increased vegetation, and increased habitat complexity, all kind of to highlight and to help visualize for Seattle residents and visitors, this beautiful thing of salmon coming back into spawn. But what they started to notice were salmon struggling in the urban creeks and streams. And looking at it, what they really noticed was mortality events, again, occurring for coho salmon as they came back into those streams. I'm just going to read a passage from Feist et al. Paper from 2011. Quote, during the first year of annual surveys on Longfellow Creek, fall coho returns were several weeks late due to unusually dry October and early November. The first significant rains in the second week of November triggered a large influx of spawners. As the rains continued over the next 2 weeks, every fish entering the drainage succumbed to the mortality syndrome, with many observations of overt symptomology during daily surveys. Fish only survived to spawn in the weeks following the mid-November storms. I'd like to preface it when they talked about fish only survived, that's some of the fish. There was mortality events continued to occur, and I'll talk more about that. So what was noticed was that mortality was proportional with presence of roads and impervious surfaces and stormwater runoff. Now, investigations led primarily through NOAA and the University of Washington at Tacoma, including this paper from Nat Schultz and Blake Feist, looked at what was really going on at the time and with the techniques that were available. And really what came from these early papers was that this was not a pathogen. It was not insecticides. There were heavy metals and PAHs, or polyaromatic hydrocarbons, in stormwater, but more so just that the mortality events directly correlated with rainfall events, impervious surfaces, and that it happened in urban areas and it was not occurring in non-urban areas away from heavy population densities. So I'm going to show a video from Nat's paper from 2011, basically looking at some of the symptoms. We have a lot of very similar videos from both out in the field and in laboratory situations. This is not a pretty video. This is a hard video for coho salmon. And what you will see is a loss of equilibrium, loss of orientation, gasping for air, and that these fish typically die within 6 to 10 hours. So this is after fish have just returned to spawn in Seattle streams and creeks. This could be in Portland, this could be in British Columbia, and after the first major stormfall or stormwater events. And as you can see, this adult, beautiful coho salmon is struggling. Fish that do show symptoms, and this fish showed symptoms within the first hour of returning into Longfellow Creek, they typically die again within 6 to 10 hours. Again, a lot of these fish, a lot of the highlight was around the pre-spawn mortality and focused on females, but the situation for urban runoff mortality syndrome affects both male and female coho salmon equally. So Julian Spromberg also had a paper in the around 2016, where they looked a little bit deeper into what was in stormwater. We knew that there are thousands of different chemicals in urban stormwater runoff, and they went about using artificial stormwater mixtures to try to elucidate what the problem really was. So they'd make artificial stormwater containing mixtures of metals, petroleum hydrocarbons, etc., but that did not cause mortality. They'd go back and use undiluted highway runoff, and that caused mortality. But furthermore, they showed a very important step, and this is part of the better management practices that's taking place with Washington Ecology, is that biofiltration, or basically putting stormwater over compost, will filter out the agent that's causing mortality. So this is a way to mitigate lethality. But again, the basic path is that stormwater comes in, it rinses something off of the streets and off of the impervious surfaces into the streams and creeks, where it ends up killing fish. So the breakthrough, and I think this is a feature of the new Trout magazine feature on 6-PPD quinone, was discovered by from Ed Kaloj's lab. And the picture I've highlighted here are two of the champions of the subject, Ed Kaloj and Jen McIntyre. And through what I think, and I think a lot of people follow this as a very heroic analytical chemistry or fractionation campaign, they went from over 2,000 different chemicals in stormwater and were able to whittle it down with this fractionation scheme to down to 4 compounds that were still causing lethality. So this was basically, you'd go through a different fractionation campaign, you'd see if the stormwater was still killing fish or not. If it was still killing fish, you would pursue with another level of chromatography. Then they got down to 4 compounds. They synthesized those 4 compounds because they were able to elucidate the structure. And they found that the causative agent was 6-PPD quinone. And 6-PPD quinone is the product of ozonization of 6-PPD. So what is 6-PPD? 6-PPD is the most abundant antioxidant or antidegradant used in vehicle tires. It's around 1 to 2% by weight of a car tire. It's ubiquitous, therefore, in the environment due to the global utilization of car tires. And it's also found in other consumer-based rubber products. 6-PPD does exactly what it's intended to do for the car tire industry and for consumers. It prevents oxidation, it extends rubber life, and again, it gives consumer safety. This picture at the bottom from the US Tire Manufacturing Association basically shows rubber with 6-PPD and without 6-PPD, where they ozonated both of those different rubber compounds. And as you can see, without 6-PPD, the rubber becomes very brittle and it starts to fall apart. It's estimated that without 6-PPD, car tires would last between 500 and 1,000 miles. So nothing like the 50 to 70,000 miles that people are now used to. An important thing that I'd like to kind of mention is that it is important, if you are in urban areas, to have your cars washed at a car wash, because they have filtration capabilities that can deal with the toxic compound. If you look at this car tire image here on the left, you'll see kind of this browning that we are all familiar with during the particularly during the summer for car tires. And this is actually 6-PPD that's reacted with ozone and that has exuded out to the surface of the sidewalls to provide protection. So this is why when you wash your car at a car wash, this gets washed off and it's collected and it does not enter storm drains. Very important. So at the time when Ed Kaloj's group discovered 6-PPD quinone, there was not a standard or a highly purified version of the 6-PPD Q. So by using that and reassessing the values, their group were able to show that the lethal concentration for coho salmon was around 95 nanograms per liter. So this is a 24-hour exposure period in a static situation. And our group was the first to duplicate those results or confirm those results, which the results were being called into question by regulators if these were true values or not. And so our group published that, yeah, our values of 84 nanograms per liter were well within the margin of error, and that around 80 to 95 parts per million is the lethal dose for killing 50% of a coho population in a laboratory-based setting. Our paper also showed that Chinook salmon and sockeye salmon were not sensitive. And this is going to be an important part later on in the talk about kind of the mystery of species sensitivity. So again, these scales are different. This is nanograms or parts per trillion. And on the right here for Chinook and sockeye, these are parts per billion. So much higher levels that would never really occur in the environment. And we show that Chinook and sockeye are not susceptible. Importantly, from a TNN paper in 2022, it listed 6-ppd-quinone as the second most toxic chemical for aquatic animal health. That falls right behind parathion. So this is an extremely toxic and potent chemical for aquatic animal health. The bar was lowered for the threshold. From Tanya Brown's lab up in BC, just last year, they showed that swim up fry. So this is about 3 weeks after the yolk sac has been absorbed, that they are extremely sensitive at that life stage, and that it is really around 41 nanograms per liter or 41 parts per trillion. So the bar was lowered. So coho salmon at this very early stage of development or early life stage are extremely sensitive to 6-ppd-quinone. But it's not just it's not just coho salmon. From the Brinkman lab and from Hiki, they were able to show that rainbow trout and brook trout are also susceptible. And again, from the Yamamoto lab, that white spotted char, trout species that's native to Eastern Asia and particularly native to Japan, they are also susceptible to 6-ppd-quinone. But there are a lot of species that are not susceptible, including, as I said, for Chinook salmon, for chum salmon, for sockeye salmon. So some of the sensitivities don't make complete sense. But the more we learn, the broader the impact. So there are 2 new additions to that list of sensitive species. In addition to coho salmon, again, I mentioned rainbow trout and brook trout, as well as white spotted char. The Brinkman lab has a paper under review right now that shows that lake trout are also sensitive. So this is very important for the Great Lakes region itself. And our lab has a paper that we are just finishing up and that we've presented at CTAC last year, or the Society of Environmental Toxicology and Chemistry, that coastal cutthroat trout are sensitive. But importantly, another group from the Brinkman lab, that is, showed that West Slope trout, cutthroat trout, excuse me, are not sensitive. So there's a big major difference between coastal cutthroats and West Slope trout. Our data shows that the coastal cutthroat trout are the second most susceptible species, period. Again, these compounds, or a general class known as PPDs that are used as antioxidants in vehicle tires, are ubiquitous. Currently, the estimates for North America is that there's about 230,000 tons of tire wear particles that are shed every year from consumer and industrial tires. I want to talk a little bit about some of the activity that's going on for 6-ppd-quinone within the USGS. And a lot of it falls again within the Biothreats program and the USGS Environmental Health Program. Particularly within the Environmental Health Program, there are 3 what are known as integrated science teams that have components that include research on 6-ppd-quinone. And on the right here, I'm just highlighting that we had a new science team dedicated to the issue of PFAS. So for the 3 different science teams that we work together on, mainly with the fishing and hunting team, they are looking at the sensitivity of invertebrate species, as well as helping the Washington Department of Ecology and US EPA establish water criteria. I'll talk about that in a few slides. We're also working to develop what's known as a national hotspot map for looking at areas within the US in particular where there might be very sensitive regions for fish species and 6-ppd-quinone. And lastly, within the immunomodulation science team, which I co-lead, we're addressing the modes of action, looking at sublethal effects, and finally, trying to find some solutions to this problem. From the fishing and hunting team, they mainly focused on mayflies and freshwater mussels. And importantly, they found that 6-ppd-quinone did not cause mortality for these species, but they did show that 6-ppd, the parental compound, which does not have a very long half-life at all, compared to 6-ppd-quinone, did cause some mortality or some deteriorated health for mayflies and for freshwater mussels. And this is work that is in progress. I think more importantly, or not more importantly, what I mentioned about the mapping effort is that there have been mapping efforts in and around Puget Sound metro area, largely through NOAA. And for the USGS, we are extending this mapping area to not only the state of Washington, but for a national map as well. And what we're mainly looking at are presence of all of the different USGS gauges in streams and creeks, and then using that in GIS technology to look at different locations where tire wear particles and tire dust could be prominent. That includes soccer fields where turf fields utilize a lot of tire wear particles, airports, looking at different traffic density, stormwater management systems, land cover, and importantly, the documented presence of fish. Again, we're building this out to a national scale. The map on the right is not for 6-ppd quinone, but from the PFAS science team. But this is an example of the coverage that we intend to do. And we have different levels of detail that we're going to put on that map. Importantly, at some point, we are going to be taking water samples from various locations to help verify if the mapping efforts for locations that we deem as being potential hotspots that could utilize better management practices will need to have sampling efforts to address those gaps. I'm having a little computer. Okay. Finally, within over at the Columbia Environmental Research Center in Columbia, Missouri, that lab has very sophisticated diluter systems for delivering real-time levels of 6-ppd quinone or other toxicants for aquatic species. Right now, the Washington Department of Ecology is suggesting a level criteria of 8 nanograms per liter, but this is still under development. At CERC, they are testing coho salmon, rainbow trout, and brook trout. And importantly, they're using a standardized fish loading rate of fish weight per volume, as well as 2 different toxicity test durations. So this is a stable flow-through situation where the levels of 6-ppd quinone are constant. And this would better mimic what is occurring within streams and creeks. The values that I presented before were all static situations. The values are likely going to be similar to what happens with the real flow situations, but we're waiting to find out how this research pans out. My group has been actively looking at the modes of action. Why is 6-ppd quinone killing coho salmon and other fish species? Right now, there are 2 leading hypotheses about how 6-ppd quinone is resulting the ill effects for coho salmon. And they focus around 2 aspects. One is around a compromised blood-brain barrier and vascular permeability. This was highlighted by a PhD student, Stephanie Blair, in Jen McIntyre's lab. And that was using whole stormwater, not purified 6-ppd quinone. She has later presented at meetings that 6-ppd quinone exerted the same type of effects on looking at compromised blood-brain barriers. The other contending theory, or potentially dual theories, are that 6-ppd quinone changes oxygen consumption rates. So basically aspects of cellular respiration. Importantly, 6-ppd quinone is metabolized by the liver, but not by the gills, where water would be passing over the gills during respiration for the fish. Our work, which I'm going to describe in a few more slides, focus on some different aspects. So we wanted to look at really the early life health effects. So we used an embryonic exposure model, and we wanted to look at different growth metrics as well as using what we call global gene regulation. So as you can see from this title, we found that 6-ppd quinone and using our what's called RNA-seq, or basically looking at the transcriptome or all of the genes that are regulated in a trout, we found that 6-ppd quinone disrupts particular pathways involved in vascular permeability. And again, I'm going to go over some of that data. Our paper that came out last year used 2 different experimental approaches, but we saw the same thing pretty much for both approaches. So again, during embryonic development, we used either 4 different pulses of 6-ppd quinone that were to mimic stormwater events for a red or fertilized eggs out in a stream or a creek. And we also followed that up with a single pulse event. What we found was that after hatching, there was not a lot of differences at the time of hatching for growth, but around 6 to 7 days post-hatch, there were significant differences in the eye area and total length of the developing alevin. We also found that at high levels of 6-ppd quinone, these are extremely high levels. Let me preface that it did cause mortality for the developing embryos, but importantly, at more environmentally relevant levels, that the chorion was able to provide a high level of protection. It was not until fish were hatching that they became susceptible. Again, the species sensitivity does not make sense if you look at it from a phylogenetic relationship point of view. So there's large differences in sensitivity between species, makes it really difficult to understand the total impact on the aquatic ecosystems. And again, the underlying mechanism is unknown. And we believe that by looking at the actual mode of action, and this is shared by other researchers, that we will better understand how, not only how 6-ppd quinone exerts toxicity, but we will be able to make better predictions about other sensitive species. Importantly, knowing the mode of action could also help us identify other potential sublethal effects. On the bottom of this phylogenetic tree, I've highlighted 2 different laboratory strains of fish that are not susceptible to 6-ppd quinone. That's the Japanese Medaka and the zebrafish. So these are standard laboratory strains of fish that are used, but they are not, we're not able to use those for this type of studies because they're not susceptible. So again, diving a little bit deeper into Stephanie Blair's paper, what she was able to show was that when she looked at coho salmon that had been injected with a tracer dye and then exposed to either stormwater or standard creek water, she found that with this fluorescent tracer technique that they could see leakage within the brain and, importantly, around the olfactory rosettes, which are important for migration. What she suggested in that paper was basically leakage occurring in the brain through leakage from endothelial cells. So endothelial cells are what are involved in forming basically all of our arteries, veins, and capillaries. If there is what's called dysfunction with a tight junction between these endothelial cells, there will be leakage from the blood. This can lead to stroke, inflammation, and other neurological disorders, which, if you think about the video I showed earlier, are phenotypes that seem associated with 6-ppd quinone. Again, I mentioned that we looked at global gene regulation, and I'm not going to go over everything about these different heat maps, but what I'd like to explain is that 6-ppd quinone dysregulates genes involved in development and in circulation. So these are looking at control fish and fish that were exposed to 3 different levels of 6-ppd quinone, where each of these little tick marks that you see represents a single gene, and each of the rows here represents an individual fish. When we look at this, it's pretty clear that when we look at showing these heat maps where yellow highlights genes that are turned on or at higher levels, that we can see a dose-dependent response. We also see a dose-independent response. If you look at this group in what's known as C5 from our paper, But importantly, again, not trying to teach about how transcriptomics works, but we found that 6-ppd quinone dysregulates or basically turns off or on genes at an alarming rate. And we're trying to investigate how that is really working. When we put all of those genes into specific pathways, again, this is a lot of data here, I apologize for that, but basically, when we look at transcriptome data, we take all of the genes and we can put them into genetic pathways. And what we found was that endothelial tight junctions was one of the top pathways that was perturbed. So again, our data strongly supports the data that Stephanie Blair presented in her early paper in 2021. As you probably know, back in 2023, California Department of Toxic Substances Control basically came out and said that there is a definite need for a 6-ppd alternative, and that needs to be addressed if the tire industry wants to continue to sell car tires within the US and in particular within California. So one of our papers is being used to address this. And again, the Science for Solutions. So we're working right now with both industry and within the Washington State Department of Ecology to find alternatives. Our second paper that had come out in 2023 not only looked at the sensitivity of coho, Chinook, and sockeye salmon, but we also mainly highlighted the use of in vitro techniques where you don't use a live animal, you can use cell lines to look at toxicity. So in that paper, this is showing 4 different cell lines from the paper. One is on the left. These are from coho salmon. The CHSE214s are for Chinook salmon. We have a cell line for rainbow trout, and we have a cell line for sockeye salmon. Again, these in vitro techniques allow us to look at toxicity without having to do lethal dose studies on living fish. It also allows us to develop high throughput screening platforms for looking at alternatives. What we were able to show in that paper was that 6-ppd quinone altered both metabolism for the cell line as well as leading to cytotoxicity at levels that were found in the environment. Again, these levels are different though. If you look at the levels we show for the cell line, they're around 6 micrograms per liter. And this is a cell line. This is not a whole organism, but we believe that the in vitro testing situation recapitulates specific aspects of toxicity of 6-ppd quinone. Finally, and probably the most important issue, are the societal effects. And that includes, in August of 2023, the Northwest Tribes petitioned the EPA to ban the parent compound 6-ppd. This again is in line with the California Department of Toxic Substance Control to find an alternative, and that finding an alternative will help aid in salmon recovery. Fast forward to November 2023, and this is probably the fastest granting that I have seen for the EPA. They granted the petition from the different tribal nations, and that the EPA intends to publish an advance notice later this year as they gather more information on the toxicity and potential substitutes for 6-ppd itself. Another highlight was that fishermen and nonprofit groups are also taking legal action. The Pacific Coast Federation of Fishermen's Association notified the U.S. tire manufacturers and basically the industry that they intend to sue. That lawsuit is progressing. And the Center of Biological Diversity notified Oregon and California state transportation agencies for their intent to sue as well. So this problem is now getting more recognition and attention. And so I'd like to talk a little bit about what a person can do, a citizen scientist or a scientist, if you witness a fish kill event and suspect 6-ppd quinone. We get a lot of emails from different residents about potential fish events or fish kill events and 6-ppd quinone. And we're coming out with a fact sheet, and we're working with Trout Unlimited to develop some criteria for that fact sheet. Basically, water can be collected, but 6-ppd quinone sticks to plastic. So we are not able to use polypropylene containers, which are commonly used for taking grab samples from streams and creeks. The preference right now is with amber glass vials. The USGS and NOAA have made significant progress in measuring 6-ppdq levels in tissues, and hopefully that method will be published this year. But importantly, if fish kill events are found, that fish can be preserved just in a freezer. So what we like to suggest is that the fish could be wrapped up, possibly in foil, placed in a manual defrost freezer. This is an important point because with a frost-free freezer, that goes through several rounds of dethaw and freezing over and over, and that will degrade 6-ppd quinone. More importantly, if you see symptoms, film it, and we're developing a fact sheet again on how to convey things that are being seen in the wild. At some point, we will likely be looking out to citizen scientists and other researchers for help in collecting water samples, again, for a national risk assessment map. And as far as our next steps, we're continuing to work with EPA to help establish guidelines. We're looking at additional species, both vertebrate and invertebrates, for potential susceptibility, and put other effects of tire wear particles on aquatic animal health. So 6-ppd and 6-ppd quinone are not the only chemicals found in tire wear particles. So we and other researchers within academia and within the government are learning more about what those chemicals, if they contribute at all to other toxic events. Finally, we're working with state and federal researchers as well as academia to find solutions to this problem. And with that, I would like to thank you for the opportunity to present this data. Again, we have a lot of partners, and again, Trout Unlimited is an important aspect of the work going forward. And with that, I'd be happy to take any questions.
That was outstanding, John. Thank you. And I really appreciate your hitting some of the questions that we knew would come up about sampling, multiple species, sampling, what you can do. I know there's going to be a lot of interest in that. So Jeff, I think you probably should oversee the questions here in the chat.
I'll try to condense
according
them,
to how you
John.
normally do this.
You know, I would say this, one of the predominant questions, we can start there while I go through rest, is what about this species and that species and this species? What needs to happen, I guess, for more research on lethal and less than lethal impacts on a wide range of species? You know, for instance, you know, we have mountain whitefish, you know, someone asked about, and others, somebody else asked about kokanee versus sockeye.
You know, those are all good questions. And fortunately or unfortunately, you know, right now we're a small group at my center, and there's a growing interest from other research groups. Our center itself here at the Western Fisheries, we have all 5 strains of Pacific salmon. That's one of the reasons why we were able to act quickly and look at species susceptibility. We are working with folks at the Colorado Department of Natural Resources about looking at some of their susceptible cutthroat species. Again, I show that dichotomy between west slope cutthroat and coastal cutthroat, one sensitive, one not sensitive. And I think it's really not until we find out the mode of action that we can really start to address on a broader scale. We definitely don't want to test species that would not make sense. So right now at the Columbia Environmental Research Center, they are looking at bass species. That is something that has come up, as well as sturgeon. And primarily, we're looking at species that are in and around areas that would correlate with this national map that we're looking at. So there are a lot of species, and it's a little bit difficult. The important thing that I try to highlight was that we're finding that there are different susceptibility or life stages for fish in general, where maybe at one particular stage, let's say an adult, they are not very susceptible, but if you go to an early developmental stage, they are extremely susceptible. So for really doing the toxicity testing correctly, one needs to go from in my point of view, from the point of fertilization to at least a year-old adult fish. So there is a list of fish that we plan on looking at, but right now we are just limited with resources.
No, that makes sense. Another common set of questions we're asking about, degradation of 6-PDQ in the water. Where does it sit in the water column, when it is in there? All sorts of things and how it interacts.
Those are great questions too. So initially, when we first started looking at this, and basically we pivoted our whole lab to focus on 6-ppd quinone, or at least my lab. It was thought that 6-ppd quinone probably would not be very stable. That was from some initial reports at conferences. It's now known that it is stable for anywhere between 14 to 28 days if you refrigerate it. So if you take stormwater or creek water that has 6-ppd quinone in it, put it in the refrigerator, it's stable and the value does not change considerably. So that's been reported by 2 different groups. Whereas 6-ppd itself, the parent compound, is very unstable. So that was shown early on when they introduced 6-ppd. It had to go through toxicity testing back in the late 70s. So this compound's been around for a while. So the parent compound is not stable. 6-ppd quinone, though, again, is stable, and it does bind to soil and sediment. And we're learning more about where it's found, not only in the water column, but within the sediment and the stability there. It does like to bind organic compounds. So sediment that's rich in mud and other organic debris will likely have 6-ppdq values. And that's why we're interested in, again, those early life stages, particularly for our group, looking at the reds or the fertilized eggs in creeks and streams, because they're directly interacting with the soil and sediment, and we're trying to determine if there's a type of concentration gradient present.
Couple questions about sampling, both sampling water quality. You mentioned kind of trying to come up with a little guidance on that, sampling the water itself, but also maybe doing, is there a way to do a necropsy on dead fish that you find? Is there even lab capacity to handle that type of, program on scale?
Yeah, no, those are, again, great questions. And as far as the capacity, it's more that the technique, as I said, is still under development, but there's been some major strides in the last month from both NOAA and within the USGS for coming up with a reproducible quantification technique for, let's say, fillets or for other tissues, the liver, things like that. People, as you can guess, are primarily interested in knowing what the values are in fillets, because what I did not talk about, and that would have been a whole nother seminar, are the potential, and I'm going to use the word potential, effects of 6-ppd quinone on human health. So it's been shown by several different groups that 6-ppd quinone, if you live in an urban area, highly urbanized area, that it's in your urine, it's in your blood, and there was a recent report that it's in cerebral spinal fluid. So this has caught the attention of the NIH and the National Toxicity Program to really investigate the potential health effects for humans. So the tribal nations in particular are very concerned, and a lot of other consumers are concerned about if this is a chemical that is harmful for not only fish, but for people as well. So we're just learning about that. As far as the capacity, that's something that we're hoping to develop with more resources. So again, it's sort of a stay tuned, and we'll update.
Appreciate that. There are a number of questions about best management practices for managing stormwater runoff. You had mentioned the compost, so there were some technical questions about how deep, how thick. Others were asking about would simple repairing buffers work, or stormwater retention. Is there, the studies, I know that work is still kind of going on, and there's going to be a lot of arguments, but for practitioners, most of the folks on this call are people who are doing, habitat restoration and are practitioners in the field. Are there general things about, mitigating and slowing that stormwater that you think, different treatments, worth the effort now while all the more research goes on, while all the more advocacy for removing it, while all the more study of a replacement chemical goes on.
Yeah, I guess I'm probably not the best person to talk about the better management practices. I'm pretty familiar with them, but what I would really suggest is that people that are interested is to go to the Washington Ecology website. They have a specific website that's focused on 6-ppdq, and they're updating it. They will be providing a significant update in June, but they also talk quite a bit about the different better management practices, including work that was recently published by the Washington Stormwater Center down in Tacoma. So I would say that those are really good resources to look at as far as the better management practices. And again, I made it a very simplified view when I talked about compost. There are a lot more sophisticated technology that are being implemented, that are actually being implemented now on different bridges in and around the Seattle metro area. But again, Washington Ecology is probably the best source to go to for that information.
Tires in the river. Can we just assume if it's a 100-pound tire that there's a pound of 6 ppd sitting and slowly being released into the water, and every tire we pull out, how many tires in a mile of river, I guess, would get to lethal levels or impactful level?
Yeah, those are, I'm prefacing this in the probably the way that's to be understood. Those are kind of terrible things to think about and questions. We don't really know fully about the full leaching capability, and tires, as you know, are used for a lot of different situations that are to enhance aquatic life. So, and there's also recommendations of using tire wear particles in asphalt itself for helping as a way of dealing with recycling of car tires. The one thing I did say is that 6-ppd, the parental compound, is highly unstable. So as it is released, it is degraded quite quickly. But there have been reports showing that 6-ppd that leaches out of a car tire can likely go through an oxidation event where it becomes 6-pp quinone. But those levels I would think would be quite low. So when I'm talking about these toxic events for stormwater runoff, that's typically those are heavily used roadways. There's a lot of debris that's being car tire dust that's being released and being swept off into the streams in a very concentrated type of format. So that is a question. And again, NOAA as well as Washington Ecology are looking at the fate of those car tires. I think a lot of the folks that do 6-ppd quinone tox will show that, random stranded car tire that's out either in a river or stream, or even like when I've been out on Puget Sound during ultra low tides, you can find them on sand flats as well. So that's something we're still learning more about. Like what kind of contribution are those car tires doing towards any toxic events if that does occur?
I appreciate it. Maybe time for one more, and probably a decent one is what the auto and the tire and the rubber industry are doing. What have they said, and how are they working with you and other partners of yours on this issue?
Yeah, so I guess one encouraging aspect is that when the TNN paper came out in early 2021, or technically December 2020, that they were alerted early and right away for that publication. They took it very seriously, and they've been working very closely with Washington Ecology as well to help address its alternatives. As I mentioned, we are working with them under a collaborative research and development agreement to help find alternatives using our in vitro approach. And there are other groups that are working with industry as well to find potentially greener solutions for 6-ppd as well. So the tire industry is taking it seriously, and I don't think it's just about the monetary aspects. They realize that this is killing coho and other fish species, and they want to correct that problem. But I cannot speak for the tire industry itself.
I am going to do one more, and this is a classic one. Lawyers always say, never ask a question you don't know the answer to. This is a question I don't know what the question means. I'm an English major, but a couple people have asked about fluorescence, Raman emission signatures, those types of things. Does that ring to you? Do you know what what they're asking there?
Yeah, I do know. So 6-PPD quinone does have a fluorescent signature. Well, right now we and other groups don't know the best instrumentation for looking at that. The signal is not a very strong signal, but it does have a particular wavelength that it emits as far as a fluorescent signature. So that is something that we have tried to measure with our standard, what's known as a TCAN or a fluorescent plate reader, but the values were not reproducible as far as establishing actual levels of 6-PPD quinone compared to the standardized techniques using mass spectroscopy. So it's a great question. It's something that we've thought about. I know from Ed Collegier's group and other groups, including over at Matt Schultz's group at NOAA, have thought about that, but right now there doesn't seem to be a viable method.
Perfect. Thank you. All right. Well,
Let me
go
chime
ahead.
in. One last
Yeah.
question I think that's relevant. John, was that would you be willing or able to share your actual slides with folks as well, or at the very least that sampling suggestions slide?
Yeah,
we could pass on?
that we can share, and as people probably know or can guess, within the federal government, we can share things that we have published and things that we have not published yet. We're not able to really share and
Okay.
distribute the information. So that is something that I think that slide is something that we're hoping to work between USGS and Trout Unlimited and probably other groups as well to find out, to develop a fact sheet. I put that slide together last night.
Yeah. Okay.
But it's
No
something
worries.
that we get
Get there.
quite a few questions on. And again, we get random emails and welcomed emails from people, from citizens that say, hey, I'm observing this event. What should I do? And I think those are really important. And again, with that national map that we're trying to develop, or that we are developing, very important.
Okay. Well, let's put our heads together and work on that fact sheet that we can get out to people as soon as we get it through the right channels. But primarily, I just wanted to say thank you so much. This has been an incredibly informative and helpful webinar, John, and you can tell by the interest of the chat and how many different questions we're getting. People are really engaged. So Jeff, I'll let you sign off here, but just wanted to say thank you. This has really been meaningful.
I will say that I see that somebody highlighted one of Ed's talks from back in February. Definitely, that's a good link to look at. Jen McIntyre has other things on YouTube, including a really nice piece that she did with the Nature Conservancy on the issue in general. And again, I think a lot of the researchers working on this issue, as well as Trout Unlimited, is trying to bring better visibility to this issue, because we still find a lot of people, even at hatchery situations, are not familiar with this topic at all. And of course, I'm biased. I think this is one of the most important issues because I'm a fish person.
Yeah.
Absolutely.
Truly important.
John, thank you so much. And everybody, thank you. We'll send the link to this recording. We'll also send the links and things that were shared in the chat as well. All of you who registered for this, you'll stay on our 6-PPD email list now going forward, so that as we do more, we'll be able to reach out with updates and asks and invites to future talks and more information. So thanks everybody for joining. Helen, John, thanks so much for your time on this.
Thank you.
Bye.
Take care, everybody.